I do hear in the background the noon whistle here at Fort Atkinson, which is my cue to say that this is Steve Larson, part of the editorial staff up here at Hoard's Dairyman. We're broadcasting here this morning from our office at Fort Atkinson, Wisconsin. And we are always happy to welcome our co-host, Mike Hutchins, down at the University of Illinois, and his partner down there, Jim Baltz, who makes sure that technically everything goes according to plan, and we always appreciate the input up here also of Patty Hirchin, online manager for Hoard's Dairyman here as part of our company. And we're really looking forward to the reproductive update here from our presenter today, and we want to especially thank the sponsor of today's webinar, Parnell. Appreciate their support very much. And Mike Hutchins down there at the University of Illinois, with those comments, I will let you go ahead and welcome the group yourself and introduce the presenter for today.
Well, Steve, thank you very much, and really it's my honor to bring back, by popular demand, Dr. Paul Fricke from the University of Wisconsin-Madison. Paul is a professional and personal friend of ours and has been on our program before and has been very, very popular. Paul grew up on a Nebraska dairy farm and row crop farm in Nebraska, got his bachelor's degree at the University of Nebraska, or Big Red as they kind of guess they go by. Then he got his master's and PhD at North Dakota State, and then in 1998 joined the University of Wisconsin-Madison staff, and where he's gone through as assistant, associate, and full professor. His appointment is 70% extension and 30% research appointment there. He's been very productive, obviously, at Wisconsin with 77 peer-reviewed papers. He has mentored 11 master's and 4 PhD students and has garnered over nearly $3 million in research funds. I'd read all his places he's been around the world. It's numerous. He travels the world, has received a number of prestigious awards here. And so, Paul, we welcome you back to our stage here at Hoard's Dairyman webinar, and we'll turn it over to you for your strategies for non-pregnant diagnosis in dairy cattle. Dr. Paul Fricke, the program is yours.
Thank you very much, Mike. Thanks for that introduction. It's an honor to be back here at the Hoard's Dairyman webinar. So I enjoy doing these, and I actually get a lot of feedback from after they happen. So thanks. So today what I want to talk about, I can talk about a number of different things, and I decided that I wanted to do a bit of an update on strategies for non-pregnancy diagnosis in dairy cows for a couple of reasons. Number one, non-pregnancy diagnosis is a key element of a reproductive management strategy on a dairy farm. And number two, there's some really interesting new technologies that have been developed that are kind of interesting that we've worked with. And so we have some polling questions and things as we go through this particular topic, and hopefully I'll be able to enlighten everyone a little bit on this area. So the first question is, why do we do non-pregnancy diagnosis? And a lot of people think it's to find the pregnant cows, but the key to early non-pregnancy diagnosis is really to find non-pregnant cows. And once we identify those non-pregnant cows, we need to couple that with a strategy to rapidly return these cows to an AI service. So I like to just tell dairy farmers that your veterinarian doesn't make you any money by telling you that a cow is pregnant. The way the veterinarian comes to your farm and makes you some money is finding those open cows and aggressively getting those cows re-inseminated. And so we're already into our first poll question. I thought I'd put this in right at the beginning, and so I'm interested to see from the attendees, what is your primary method of non-pregnancy diagnosis? And so the 4 different choices we have are transrectal palpation, transrectal ultrasonography, blood test, or a milk test.
Well,
so
Steve,
I see that people are starting.
Okay, well, at Hoard's Dairyman Farm, it would be ultrasound, primarily.
I'm going to vote for the first one, though, Steve. I just think we...
Are you?
Yep, we'll see on that.
Okay.
I'd go number 1. Not saying it's the best one, but that's where I would probably go.
Yeah, well, we have, in fact, Mondays are our herd check days, and so Dr. Rick is probably getting ready to face the cows as they come from the noon milking right now as we speak.
Well, let's go ahead and close the poll. We got nearly 2/3 of the votes in here. I think a lot of Republicans a little slow this morning. And Paul, what do you think of the results? And do you agree with the results?
Yeah, I mean, I think the results are interesting, and this is what's happened over the years, is that palpation actually came in second to ultrasound. So 40% of you said that transrectal palpation was the primary method for non-pregnancy diagnosis. 49%, or just under half, mentioned transrectal ultrasonography. And these two methods, we'll get into this, are what I call direct methods. The two newer methods, the blood test and the milk test, came in at 9% and 2% respectively. So interesting for me to see just kind of where we're going with these newer technologies. I know they're being used out there, but so interesting. 40% transrectal palpation, 49% ultrasound, 9% blood test, 2% milk test. Okay, so that launches us into I want to go through and talk a little bit about methods for non-pregnancy diagnosis in dairy cattle. And as I said, the first category that I would put these different methods into would be what I call direct methods. And so with a direct method, you're directly detecting the tissues and or associated fluids of the conceptus either manually or via electronic instrumentation. And so primarily what we're talking about here is ultrasound and palpation. Now, of course, as you said, Mike, I've been around the world quite a bit. I've been to several different countries, and so we're just going to fly through a few of these different methods for non-pregnancy diagnosis. I call this first one the wait and see method. So, you know, if you see the calf kind of coming out at you, like this cow in the lower right-hand corner, you're pretty confident that that breeding 282 days previous to that was successful. And so, obviously, this is not a very aggressive way to determine reproductive efficiency and figure out whether or not cows are pregnant or not. Gestation length in Holsteins 282 days, or about 9 months. You can see the cow up in the upper left-hand corner there. She's definitely getting ready to calve there. So this is not a method that I would really recommend, the wait and see method. Oops. Another method that I run into still, I grew up on a dairy farm, as you said, Mike, in Nebraska. My grandfather was the chief calf bumper on our farm, and he would love to. We had a 2x2 swing parlor. You stood in a pit in the middle, and the cows on the that stood on the left side of the barn, you can see this kind of tiger striped cow here in the left-hand panels right here. You can see the presentation in this figure to the right here. You can see how that calf lies in there. Because the rumen is on the left, you know, that calf will be to the right. So you just get your hand in there, the heel of your hand in there, and you can bump that calf. And so the problem with that is it's only accurate beginning about 180 to 210 days after an AI, or 6 to 7 months. So again, it's not an aggressive, it's not a really new method for non-pregnancy diagnosis. Nonetheless, Grandpa just loved to bump those calves when they came into the barn. So let's get into the two that are probably being used quite a bit out there. The first one is transrectal palpation. Really, I think this is accurate beginning about 35 days after a breeding. Data would show that if you're trying to do palpations much before that, they get quite inaccurate. This is an old method. Actually, these two fellows here, this L.E. Casta was at the University of Wisconsin, and back in 1948 published a paper on a manual method for diagnosis of pregnancy in cattle. So this is really, veterinarians throughout the years have been taught how to do rectal palpation, and most of our veterinarians are spending a lot of time during our herd health doing that. You can see how fun and easy it is here in the upper right-hand corner. One of our students here doing a rectal palpation. I actually found this cow, and this is nice because you can actually see where my hand is and what I'm doing inside of that cow. But nonetheless, you know, 40% of you said that you're still using transrectal palpation for non-pregnancy diagnosis. And I would say that this method then, transrectal ultrasound, is one that I worked on quite a bit when I first came to Wisconsin about 20 years ago. Did a lot of training of veterinarians to do ultrasound, and my prediction was that over time, ultrasound would start to displace transrectal palpation as the primary method for non-pregnancy diagnosis. And based on the poll, that's exactly what's happened. And so those were the two most popular, but there's more people out there taking advantage of ultrasound than there is of palpation. And so here you can see in the left the veterinarian who is doing an ultrasound outside on a group of heifers, and you can see that he has this heads-up display, and this is the sort of thing you'd look at in the right-hand side here. You can see this. Actually, if you look at the name on the upper left-hand corner, Carol Fricke, that happens to be my wife. So this is my son before he was born. But that's just to say that ultrasound was first used in humans. It's a very interesting technology. It's a very accurate technology. You can see the viability of the fetus. You can age. You can do many different things with this. And I think a lot of veterinarians now are using transrectal ultrasound. Notice that I say it's accurate beginning about 30 days after insemination. I'll come back to this. Originally, I think we thought that we could do ultrasound a little bit earlier than this, but I've been telling people to wait till at least day 30 to do ultrasound. So before I talk about some of the newer methods of non-pregnancy diagnosis, I wanted to give everybody a feel for the costs associated with non-pregnancy diagnosis, and this would be cost per cow. And so this slide is probably about a year old, but I called around to many of the different veterinarians in Wisconsin that I know. And so you could put together a grid like this yourself as well. So what I did is for palpation, basically $120 to $130 per hour is what a lot of veterinarians are charging for their time to do that. With ultrasound, it's a little bit more expensive, obviously, because you have to pay for the equipment. And so cost per hour for a veterinarian to do ultrasound would be between $150 to $160. So if you know your cost per hour, what it comes down to then is how many cows you can actually roll through per hour on a farm. And so this number is going to be dependent upon the size of the farm, how well the cows are set up when the veterinary gets there, whether you're working in a palpation rail or going down in cows caught in a headlock. And so I would say that many of our farms in Wisconsin would be on the lower end here, maybe 40 to 60 cows per hour. Moving to the right, a lot of the dairies probably in the southwestern United States, some of our larger farms here in Wisconsin, you could potentially get to 100 cows per hour. You've got to be moving through them. It's not impossible to do, and you can do it quite accurately, but it's just a matter of having those cows lined up. And so you do the math and you can figure out that the cost of per cow of doing non-pregnancy diagnosis can range from, $3 to $4, $2 to $2.67, $1.50 to $2, or as low as $1.20 to $1.60. And so I just want you to kind of get these ideas in your head of what this is going to cost, because when we talk about the newer methods for non-pregnancy diagnosis, you'll see the costs of those methods. And in fact, the costs of some of those newer methods are based on this kind of a calculation. They're going to have to be competitive if they're going to displace these older methods. So here's our second poll question then. And the question is, at what days after an insemination do you conduct first pregnancy check? And so what I'm asking here is cows are inseminated, and then what day post-insemination is the earliest that you would do a non-pregnancy diagnosis? And you can see less than 25 days, 25 to 31 days, 32 to 39 days, 40 to 47 days, and greater than 48 days. So it looks like people are rolling.
People are voting quickly there. I think in the case of our farm, it would probably be 32 to 39. Mike and Paul, but let's see what the votes coming in pretty fast there.
Yep.
60% voted already. Yeah, that's great.
I think all the Trump votes are in already, to tell the truth.
All right, yeah.
Well, I can't vote. I have no idea what we're doing on our farm, so mum's the word on this end here.
Yeah.
Well, I think we're going to close this off. We're at 2/3 vote. Paul, any surprises?
Yeah, I don't think necessarily surprising. It actually kind of turned out the way that I thought it might. Nobody is less than 25 days, which is really good because we really don't have a way to do that. That's very accurate. The most popular would be, Steve, where you talked about 32 to 39 days. That's what I've been advocating, especially if you're using the two direct methods that I've just discussed. Second most popular would be 25 to 31, which indicates to me that there are still some people out there doing ultrasound that would be pushing this down. And then you get down into these last two categories. 13% said 40 to 47 days, which in my opinion, that's too late. You're losing a lot from reproductive performance when you let cows go that long before you diagnose them open. and then there was just 2% more than 48 days. And of course, not very many people are going to be that late. But anyhow, it's interesting spectrum. Everybody across these lower categories, nobody less than 25 days. So very interesting. So I want to try to convince everybody, based on that poll question, that I think that you can do pregnancy diagnosis too early. I'll set this up. This is an experiment that we did here at the University of Wisconsin, and cows received a timed insemination on a Thursday. And so in this particular experiment, we were working with a farm. Everybody was set up on a timed insemination on a Thursday. And at this time, we were looking at a double Ovsynch program to resynchronize the cows. Now, I don't recommend that because the interval between inseminations is too long. But what it did, if I put this up, you essentially are going to treat with GnRH 22 days after this insemination, and it forces you to do an ultrasound on day 29. Now, the reason I'm showing everybody this is because early on, I would have said day 29, it would have been fine to do an ultrasound. In fact, we would have advocated 28 and 29 days. That was what ultrasound was for, so we can do these early non-pregnancy diagnoses. And so the way this was set up, every single cow was set up to be pregnancy diagnosed exactly on day 29. Notice the interval between breedings, 49 days. That's where this kind of a strategy really falls down. And again, I'm not recommending this resync strategy, just showing you the context of the experiment and showing you that all cows were diagnosed for non-pregnancy using ultrasound on day 29. So here's the results, and we published this in a book chapter on non-pregnancy diagnosis that just came out this year. So all the cows were checked 29 days after timed insemination. This was all done by my graduate student who was a veterinarian, very good with ultrasound. We had 1,116 cows, and what I had my grad student do was basically say what he was looking at when he made this diagnosis. And we would say that you really need to see the embryo with the heartbeat, the corpus luteum on the ovary on the same side as the pregnancy, and fluid in the uterine horn. And so only 68% of these cows could he call pregnant based on the fact that he saw an embryo with a heartbeat. So think about that number. That's not a very high number. So you're only seeing that embryonic heartbeat with the fluid in the embryo and the CL in 68%. He had to diagnose 29% of these cows pregnant based on the fact that they just had a CL with fluid in the uterine horn. These were questionable pregnant. These are animals that had some fluid in the uterine horn, couldn't see the embryo, but it was less than the normal amount of fluid. So what I'll put up now then is basically we do rechecks. So you can see day 39 the cows were rechecked, day 74 the cows were rechecked. Let's look at this bottom line in red. This is the loss from 29 to 74 days. And the point I want to make is this. If you see the embryo with the heartbeat in addition to the corpus luteum, pregnancy loss from 29 to 74 days was only 9%. And that's very close to what I would consider the biological loss would normally be during this particular time period. Look at what happens to loss if you make these diagnoses based on fluid in the uterus alone. In other words, can't see the embryo. 28% pregnancy loss. In these animals, obviously the questionable pregnant ones, less than normal amount of fluid. What's happening in these two groups is you're looking at pregnancies that are in the midst of being lost. And so the problem with this is that you cannot take action on these animals as open animals. So essentially you're going to call them pregnant because you can't finish the protocol, you can't get prostaglandin, and then you're going to see a very high rate of pregnancy loss. And I started seeing this kind of a trend in our industry at least, where people were seeing very high pregnancy losses when people were trying to do these pregnancy checks too early. So just a bit of a warning on that. I think you need to get out to day 30 to 32 with ultrasound to get this number where it needs to be and not calling these cows pregnant. And this just shows you these are some cross sections of what you're actually looking at. So on day 25, I'm basically outlining the outer part of the uterine horn, and then this is all the fluid that's there. Each of these boxes, by the way, is 1 centimeter. So you're just looking at a very small amount of fluid in a very short period of time. In fact, in 7 days, this is the lumen of the uterus, and now you can see the amnion and you can see the embryo itself. And the difference here is that when a pregnancy loss happens in this case, this tissue, there isn't much there, that embryo will die, and it just looks like fluid. There's a corpus luteum there because there's still something in the uterus. In other words, this kind of fools you into thinking the cow is pregnant. These, when you get to day 32 to day 39, when these embryos have died, you can see that the tissue becomes very unorganized, and you can actually call those pregnancy losses. So just a very short period of time, there's a tremendous amount of difference in what you're seeing with ultrasound with the non-pregnancy diagnosis. Need to discuss a little bit about pregnancy losses in lactating dairy cows, and of course I've already kind of brought up this concept. There's a lot of pregnancy loss in lactating dairy cows. This is one of the only studies that I'm aware of that has really tracked loss from day 28 all the way till calving and generating this particular curve. And so what you can see, these animals had to be pregnant with an embryo and a heartbeat on day 28. So you can see from 28 to 42 there was 11% loss. From 42 to 56 there was an additional 6.3% loss. And then the losses get a lot lower out here. And notice the inflection point on this curve is about at 70 days. Notice that the total loss was about 1 in 4, about 25%, or about 1 in 4 pregnancies is lost from day 28 until calving. But the shape of this curve, we can use that to our advantage to set up a reproductive management program with regards to non-pregnancy diagnosis. And so just a couple more data sets. Again, one of my grad students in 2012, just some of the loss from 29 to 39, just under 9%. 39 to 74, just over 7%. So from 29 to 74, about 15.4% loss. This is another study then. Oops, I didn't hit that. Okay, so what you would want to do then, this would be very typical of farms that I see. I always recommend a minimum of 3 non-pregnancy diagnoses. The first one on this farm is happening essentially around day 32 to day 39. You can see that these animals are first checked here. There's a first recheck that happens, and this is going to be placed right around the time of the inflection point of that curve. I would even say day 68 would probably be better. This is a little bit later than that. And then there's going to be one late in gestation here just to make sure that you're not drying off non-pregnant cows. So I would suggest a minimum of 3 non-pregnancy diagnoses within the scheme of a reproductive management program. Okay, so those are some of the direct methods, or the 2 direct methods that are most popular. There's some newer methods now that are out that we just need to discuss, and these are what I call indirect methods because they're basically measuring reproductive hormones at specific stages after an insemination, or detection of conceptus-specific substances in maternal body fluids. And there's a couple of different strategies that we can look at. So this middle picture is just a reproductive tract from a dairy cow. I've blown up the ovary on the left, and you can see the 2 structures. You have the follicle on the left, you have the corpus luteum on the right. The corpus luteum produces a steroid hormone called progesterone. And progesterone changes throughout the reproductive cycle, and we can use those changes to determine whether or not a cow is pregnant. So just an example here, the upper left-hand panel, this is an experiment that I did, and each little dot here represents a Monday, Wednesday, Friday, and I just took a blood sample from these cows. These cows were synchronized to receive a timed insemination on May 1st. And so you can see this animal synchronized, she ovulated, progesterone came up, she did not get pregnant, so the CO regresses, progesterone comes back down again. She had another cycle, she would have come into estrus, she would have ovulated, formed a new corpus luteum, and so you can see this cow is cycling. The bottom picture then shows a cow that she received a timed insemination on May 1st, progesterone comes up, and progesterone stays up. The power of looking at these is the repeated sampling. And that's where the power in this kind of thing goes, because I didn't have to really put my arm into this cow to determine whether or not she was pregnant. I just want everybody to kind of imagine a technology in the future. If we could measure milk progesterone in line, even 3 times a week, daily would be better, we could radically change the way we manage reproduction on our farms. Now, there used to be milk progesterone tests that were out, and what they would say is that, look, if it's low 20 to 24 days after AI, this cow is open. If it's high, then this cow is pregnant. Now, that sounds all well and good, but the problem comes in with the low diagnosis. The low diagnosis is actually very accurate. The problem comes in with the high animals, and I just want to hit that concept on this slide. So this is an experiment we did. We had 55 cows that we set up for a timed insemination. And then we did an ultrasound on day 29 or day 30. We could classify these cows. 29 of them were pregnant, 26 of them were open. The pregnant cows, progesterone comes up and stays up, just like the textbooks say it should happen. But we could easily categorize these open cows into two groups. And so 65% of the animals had a normal time of luteal regression. In other words, these cows read the textbook, they didn't get pregnant, they regressed their CL. These cows would have been back in heat around 21 or 22 days. But 35% of these animals had extended luteal phases. In other words, these cows would have tested pregnant based on high progesterone, but they didn't stay pregnant. We have another data set that I won't go into today. We had a tremendous amount, about half of the cows had a normal luteal phase. The other half of these animals have all these extended luteal phases, and what you're looking at is early pregnancy loss. These animals are pregnant long enough, they maintain the corpus luteum to keep progesterone high, but then subsequent to that, then they lose that pregnancy. And so this really changes the dynamics of what's going on and how we think about non-pregnancy diagnosis using these. But again, if I could test daily in milk, then I could actually see this happen. I could see when a loss happened. And it just so happens that there is a technology out there. The DeLaval Herd Navigator is a system that's present in many different parts of the world. I believe there's one test unit in the United States, and it's here in Wisconsin, not far from Madison. But what this is a robot that tests three different things. It tests milk progesterone, as I've just described it. It looks at mastitis through LDH and ketosis through milk BHBA. So there's something there, Mike, for the nutritionists, for the milk quality people, as well as for the repro people. But this is really an interesting technology. I think they're going through some things with the FDA. It's not approved here, but there are systems in Canada and in Europe, and it's very exciting to see this kind of idea starting to actually be put into practice. I think this is only going to get better as we move forward. It's the technology that I think is really going to change the way that we deal with reproduction in the future. Okay, so that's progesterone. I want to talk a little bit then about use of pregnancy-specific markers. And I show this graph because for many, many years, most of you out there know, in humans, there are these tests, and I'm just showing you a picture of one. They're 99% accurate. It's a simple laminar flow, lateral flow test. This is in urine, in fact, and you can see if you got the two lines here, you're pregnant. If you've got the one line, you're not pregnant. And so we've been able to do these very simple, inexpensive, highly accurate tests for pregnancy in humans based on this marker. It's called human chorionic gonadotropin, and you can just see how it looks. It comes up very high, and then it comes back down, but it's there, it gets into the urine, it's excreted in urine, and we can measure it extremely accurately. People have always asked, we can do it in humans, why can't we do this in cattle? And the reason we can't is that ruminants do not have a chorionic gonadotropin. The only species that have a chorionic gonadotropin are equine, or horses, and humans and primates. So we can use this very accurately in humans and primates, but we have to look for something else. There's just no chorionic gonadotropin. And so the search was on for many years to look for some kind of a marker that would be accurate. And this is work from 1982, and this third author, Garth Sasser, was one of the originators. They basically found they found two different proteins associated with pregnancy. One was pregnancy-specific protein A. It was alpha-fetoprotein, and it wasn't a novel protein. So then they turned to this second one, pregnancy-specific protein B. We call it PSPB. And it was kind of a novel pregnancy marker produced by the bovine conceptus. So once you have a novel marker, you can basically make an assay and you come up with a pregnancy test. So this is Dr. Sasser's, one of his second papers. This is 1986. They developed a radioimmunoassay where they could measure PSPB. And this is the profile. You can see somewhere here about day 30 or so, you see it come up. And this is really important to pay attention to. It comes up. It comes back down, does this little dipsy-do here, and then it gets very high throughout gestation. So if you're going to use this test, there's two things you have to think about. Number one, the earliest pregnancy check you can do is about 28 to 30 days after insemination. These numbers are kind of specific for the different tests, so what I would recommend, just read read the material with the test to figure out what day you can do it the earliest. And the second thing you have to pay attention to is once the cow calves, the calf is gone, but the maternal blood is full of this PSPB, and it takes a while to get it out of there. In fact, it takes up to 60 days after calving for the cow to test open. In other words, if you test a cow within 60 days of calving, she may test positive just from the PSPB from the previous pregnancy. So cows have to be more than 60 days post-calving, and they have to be about 28 to 30 days after AI. And if we've worked with these tests, which I'll show you in just a minute, and they're very accurate if you hold true to those two constraints. There's another name for these things. We call them PAGs. They're pregnancy-associated glycoproteins. It ends up that PSPB has basically been identified as part of a larger family. PSPB might be PAG1, and then there's more than 20 of these things. So there's all these other PAGs. Nobody knows exactly the biological function of these things. They're inactive aspartate proteinases. So the function of these things during pregnancy is really unclear, but we can use them as a marker to determine whether or not cows are pregnant. So this is just a cross-section, a cartoon of the cross-section of the placenta. You have the maternal side here, you have the fetal side. These green cells, these binucleate cells, originate in the fetal side. It takes some time to get them to migrate to the maternal side. These granules inside are basically filled with PAGs that get excreted into maternal circulation where we can then measure it. So basically now there's a whole group of these different tests, and I want to just show you some of the different companies that are working with this. I think these are the three companies that still do the blood test. So I just went onto the web and pulled some stuff down. So you can see until you train them to pee on a stick, of course this harkens back to the human test, right? PSPB does not get into urine, so we have to do a blood or a milk test, okay? So the first tests were blood. So Bioprin is kind of the first to market. It's an ELISA. They set up labs in the United States 28 to 30 days after AI. And look at the cost, $240 to $275 per sample, and that's the cost of the test only. So that doesn't include shipping. It doesn't include the needles and needle holders and all that kind of stuff to collect the sample. So you'll notice that cost is getting into kind of the mid-range of what it would cost to do an ultrasound or a palpation. So BioTracking is one company. Another one is called this DG29, marketed by CRI in the United States, and there's lots of good information up there. You can pay for the test only or the complete test. So to get a feel for what these things cost for all the costs associated, 24 tests for $3.54. The more tests you buy, the cheaper it is. So you're getting that down to about $3.30 per test, and that's everything. That's the test analysis, blood tubes, needles, needle holder, shipping containers, shipping costs, and everything. So again, this is creeping up into maybe even the higher cost, but it's still going to be competitive with an ultrasound or a palpation. And the last one is a company called IDEXX, and IDEXX came out with their bovine pregnancy test a number of years ago. And again, $250 to $350 per test, and again, that's the cost of the test only. So these are very comparable tests. They're different, different companies that are marketing it. Everybody got their own PAG, essentially. They built antibodies to it, and then they can market and make these tests. So I just want to show you what this looks like. This is a test that I worked with. This is actually the visual read pregnancy test from IDEXX. So it's an ELISA plate, 96-well plate. And this is a standard curve. We took serum from pregnant cows, pooled it, and you can see that we just diluted it down, so you can see that's what it looks like. And then right here, what you can see is these are duplicate samples from the same cow on the different days post-insemination. So you can see no color change at 20, none at 22. Day 25, it starts to light up. Day 27, even more so. And you can see this cow got pregnant. This cow here obviously did not get pregnant. And so it's a colorimetric test. You don't need a reader to read it. It's pretty neat technology. They're marketing this to veterinarians, but places in the world where you can't get ultrasound and those kind of things, I think this test really has a lot to offer. And we've worked with it in a research context quite a bit. So with that, I know there weren't many people using the blood or the blood test, but for those of you that are using a blood test, The poll question is, are you using the Bioprin test, DG29, IDEX bovine pregnancy test, or the IDEX visual read pregnancy test? So let's see what people say.
Well, the lines are open. Steve, are you using that on hordes at all?
No, we're not. So I'm not going to weigh in on this, but people are voting here rather slowly.
Yeah, but if you remember, there was only about 10 to 12% of the total people that were using these tests. So we probably have it almost done here.
Not applicable might be the.
Yeah, I should have put. Well, I kind of said if you use a blood test, right?
Yeah, that's okay. Yeah, you got it right.
Let's sign her off. I think we're done. Okay. Paul, you're right. What do you think?
Okay. Yeah, no, very interesting. 58% of you used Bioprin, and again, that's kind of the one that was out the gate first. They really established themselves with labs all over the place. Probably the one that you can most conveniently do, with a veterinarian. And so 58% Bioprin, 11% DG29, 11% the IDEX bovine pregnancy test. But I guess one thing that surprises me, 21% the visual read pregnancy test. I love this test. And you can run it on a farm. It's not cow side, but you don't have to send those samples away. If you have a clean room on the farm, you can run this test very easily. And they've got a new version of this thing out where I think the time to run this test is now 20 minutes or so. The old test used to be close to 2 hours, I think, to do all the incubations, and this is getting down to 20, 30, 40 minutes, somewhere in that range. So really kind of interesting. I do think that these tests are popular, and they're going to continue to stay so. Okay, so I want to just go through some of the work that we've done with these tests. And again, this is newer technology, and so kind of interesting to see how these things can be fit into a management strategy. So the IDEX test was actually, the technology behind that was actually developed by Monsanto. And this is a study that we did with Monsanto early on when they were developing that test. And a couple of my grad students, Elena's doing an ultrasound, and Ryan's pulling a blood sample for the PAGs. And so we can essentially use ultrasound as the gold standard to determine the accuracy of a PAG test. And so basically, they came to us and they said, if you had a technology like this, like you could take a blood sample anytime you wanted, and you could structure that around a strategy to re-inseminate cows, how would you do it? And so I said, well, what I'd want to do is, here we have a first time insemination on a Friday. And so day 1, day 2 is Sunday. So the days on Sundays, I've marked how many days post-insemination that is. Then I said, well, I'd want to be aggressive. And so this is day 23, 24, 25. So day 25, I'm going to set up cows to receive GnRH. Now, we don't know whether or not they're pregnant here or not. GnRH is relatively inexpensive here in the United States, and so you can set up cows with GnRH. It's not going to hurt the pregnancy. And then day 27, we'll pull the blood sample for the pregnancy-associated glycoprotein test, and we'll do an ultrasound at that same time. And so we're using ultrasound as the gold standard for the PAG test on day 27. Now, if you remember, this is early to do an ultrasound, and so we had to really work with the data to make sure we knew for sure that they were pregnant with ultrasound, and then we could assess the accuracy of the test. You have to send the test away to a lab to get done, and then you basically get emailed the results. You have to know the results by the time you roll around to this prostaglandin. Pregnant cows will drop out. Non-pregnant cows are going to get prostaglandin, 2 days later GnRH, and so now we're going to have a nice interval, a short interval between timed inseminations. And so this just shows you kind of how we did this. Basically, the time from sample collection to receive the outcomes was 36 hours. So students are out early in the morning doing their thing, taking the blood samples, doing ultrasound, come back to the lab, spin down the samples, and ship them, overnight express them to St. Louis. This is where they ran the test, and then they get those results, and they would email them to us. There's the Animal Science Building at the University of Wisconsin. And then we would essentially get that information out to the farm. And so turnaround time is about 36 hours. And I think one of the things that limits these tests a little bit is they're not cow side and they're not on farm. And so, I think they're working on iterations of the test for that. I was actually surprised at the uptake. In fact, Bioprin was 58% of you who are using these tests were using Bioprin. So this is the kind of thing you have to go through. You have to collect the samples, ship them away, and then get the results back. And cow identification becomes incredibly important. You have to put the right cow number on the right tube and make sure that human beings read the same numbers, and be clear with your writing and all that kind of stuff. And then that information has to stay attached to each individual sample through this whole process until you get back. So a lot of logistics involved, but a lot of people out there using this. Now, what we did from this study is we basically calculated, there's a number of different things you can calculate. I'm not going to go through all of these, but the two most important from an economic standpoint are the overall accuracy of the test, that's 95%, and the sensitivity, which is the proportion of samples from pregnant cows with a positive PAG ELISA. In other words, this is how accurate a pregnant diagnosis is. And usually when I show these numbers, people look at that and say, 95%. Well, it's not 100%. And the thing you have to ask yourself is 95% good enough? With any analytical test, you're never going to have analytical tests that are 100%. So nothing's 100% accurate. And so the question you should be asking yourself is 95% accuracy, 95% sensitivity good enough to economically run this particular test? And so this is a study that we published back in 2013. And so we did an economic analysis. This is using a statistical economic model. And so to be economical, the sensitivity of the test had to be greater than 94% when you use 24 days after insemination, or 96% when used 31 days after insemination. We're right between these two numbers. And our conclusion in this particular paper was the technology as we have it now is economical. And so I think a lot of people are using these tests with great success out there in the dairy industry. So that's the blood test. And so what happened a number of years ago, in fact in 2012, is that IDEXX came out and they said, well, now we've modified our test so that we can use it in milk. And which is an interesting idea because a lot of farms are doing DHIA testing. And so what they did is they set up with they partnered with the DHI services around the country, and they set up the milk PAG ELISA in those labs. So essentially what you can do now as a dairy farmer is when you send in your milk samples to do your milk testing, you can check a box that says, I want a pregnancy test done. Okay, and so you can see the manufacturer suggested retail price of that is $3.50 per sample. The range $3 to $5 depending on who you're working with. Now, one of the things that it's a neat idea, but here's what I always say about this: for a good reproductive management program, you have to be able to detect non-pregnant cows on a weekly basis. Otherwise, cows go too long being open. And so most people test with DHIA monthly. And so, we're still kind of working on how to maybe get this to work right. One of the original ways they marketed this test was as a recheck and at 60 days. And let me just roll through some of the data that we have on this. So this is a paper we just published last year. And what we did is we had a group of cows and we had blood sample and milk samples and ultrasound from the same group of cows. And so let me just go through this. PAG levels change associated with stage of gestation, parity, pregnancy loss, and milk production. And these changes will then, in turn, determine when we should do the test and when it's going to be most accurate. So here's how we did it. We had cows on these different days in gestation. We did an initial ultrasound of 128 cows on day 25 and day 32, and then only pregnant cows went on. So weekly we had these ultrasounds. We made sure that they had an embryo with a heartbeat, and then we had blood and milk samples from these cows. So we could do sensitivity and specificity on day 32 because we had a mix of pregnant and open cows. Keep in mind these are only pregnant cows out here, so we couldn't do those calculations. So I think what I have first is the sensitivity specificity on day 32. Yeah, so this is what we published. So if you look at the plasma results, the blood test sensitivity was 100% on day 32, and in the milk samples it was 98%. those are incredibly high sensitivities and make the test extremely accurate on day 32. And I want to show you why that's the case. Actually, I'll show you this first. So we had 137 cows that were pregnant, or 57 out of 137 were pregnant on day 32. A couple of cows were diagnosed with twins, and these cows were removed from the analyses. We wish we would have had more twins because it's really interesting. Cows with twins have higher PAGs than cows that don't. And then there's always going to be pregnancy loss. And what I want you to remember is that in the field, you can't take those cows out. You have to deal with the fact that there's going to be cows that are going to be losing pregnancies, you know, dynamically as you test for these animals, or test these animals for pregnancy. So 7 out of 55 cows lost a pregnancy, and we could look and see what happened. This is plasma PAGs, and these are milk PAGs on the same group of cows, so these 7 cows. Notice that every one of these cows would have tested positive or pregnant at some point in time, and then they lose their pregnancy, and it takes 2 to 3 weeks to get those PAGs back down to where they would test open. So we have the luxury in this data set, we could take those animals out of the analysis, which is really important because that pregnancy loss confounds everything. And so what we ended up with was a group of cows that stayed pregnant. Okay, so no pregnancy loss in here. So now we can look at what do PAGs look like from 25 to 102 days. This is essentially the first trimester of gestation. And so this upper graph is all cows. Day 25 is too early to test. These are the upper and lower thresholds. If cows fall in between, they're rechecks. If they fall below, they're open. And these are just cutoff points of the assay that they use. Look at the pattern of PAGs, and if you remember back what I showed you with the PSPB, it's exactly the same thing. Too early on day 25. They get very high in pregnant cows on day 32. This is why the test is so accurate. And then we don't know the reason why these PAGs do this. Even in pregnant cows, they come back down, and in fact, pregnant cows will get into a situation where they either test recheck or open even during this particular time, and then it comes back up. So these bars over on the right side correspond to that. So day 25, these are pregnant cows in the clear bar that tested pregnant cows that tested recheck in the hatch bar, and pregnant cows that tested open in the black bar. Obviously, the black bars are the worst because you would essentially be terminating a pregnancy if you would give prostaglandin based on the test. Notice that on day 32, the test is 100% accurate for diagnosing pregnancy. When these PAGs come back down, you can see that there's a lot of rechecks generated, and a recheck, you know, what's your management decision if you get a recheck? You can't give them prostaglandin and finish a protocol, for example. You assume that they're pregnant, so you have to retest them. So the rechecks even get problematic here. So the other thing I'll show you here, first lactation cows always have higher PAGs than older cows do, which is very interesting. So the test is going to be more accurate for the first lactation cows than it is for the older cows. And those bars over there. Now this is milk PAGs. So the previous slide was the blood test. This is the milk test. You have about 2-fold lower PAGs but the same pattern, and they just have the test a little more dialed in. You can see the pattern here. It comes back down, and so very accurate on 32 and 39. A few pregnant cows will test open, which is really a bad thing. You get some rechecks in there. And so we basically concluded from this, I mean, if we could choose when to do a test, day 32 is just the best time to do either the blood or the milk test. I would personally try to avoid these later times. Now, keep in mind that, you know, there's a lot of pregnant cows that test pregnant here, so that's fine, but it doesn't take many cows with a wrong answer to start really messing up the sensitivity and specificity of these results. And the last thing I'll show you here is this. And so we basically looked at the impact of milk production on PAGs. So this is milk production on milk PAGs. The first lactation animals, you can see, as milk production goes up, PAG levels are lower. In the older cows, you can see they're given more milk, same kind of slight negative relationship. And we looked at this graph and we said, aha, it's a dilution effect. The more milk they produce, they're just diluting the PAGs in milk, and that's the problem. It ends up that's not the problem because these are the same cows now in plasma. That negative effect is there in the first lactation animals and in the older animals. This is not a dilution effect. It's probably either a production or a metabolism issue. And so this relationship, we have some really new, neat data that I'm not going to get in today, really looking at this and seeing what is going on. We think that in higher producing cows, they have less progesterone, and the embryo actually grows a little bit slower and decreases PAG levels. So we concluded then all these things affected these PAG levels. I would do a preg check around day 32, coinciding with those early peak PAG levels of pregnant cows. And keep in mind that because there is pregnancy loss, the cows have to be retested at greater than 74 days, or you could even palpate them later too. I mean, you just have to do that pregnancy recheck. So with that, I think, Mike, I finished exactly on time at 12:50 here. Again, I'm from the University of Wisconsin-Madison, and we have a lot of fun here in dairy science. And I do have a website if you guys want to visit that. The URL is down below. If you click on this extension link, you'll get a lot of the information that I went through. In fact, this slide set is probably up there somewhere. So with that, I'll turn it back over to you, Mike, and we'll see if there's any questions out there.
Well, Steve, I guess you want some comments here before we get into Q&A?
Yes, I will. While you organize the questions and Paul takes a breath there, I want to thank Dr. Paul Fricke very much for a wonderful presentation. We need to advance the slides there, Paul, if you will.
Yep.
That for us. Thank you very much for your great presentation. Some really interesting look at helping us sort through all those options. And we want to thank Parnell for their support of the webinar today. Reminder for those of you that are with us, you'll be receiving a survey here in a few days about your impression of this webinar, and we would really appreciate your participation. Just takes a couple of minutes for you to do it, and it's very helpful for us. And of course, this webinar, after a few days, and all of our previous webinars are available. They're archived, free, and ready for your viewing of your at hordes.com. Go to our webinar section of our homepage. And also, there actually would be just a few minutes left for anyone who would want to download the PDFs of the PowerPoints that were used today. You could still do that here in just a few minutes. Then looking ahead, if we advance one more, there we go. The next webinar, hope you can join us, June 13th. Jack Rodenberg from Ontario will be talking about barn designs and management of cows in the robotic systems. He's with been with the Ontario Ministry of Ag and also Dairy Logics consulting firm. And the sponsor of that time will be Lele. And we look forward to that presentation very much. And then spinning ahead just a little bit further, July 11, Mike Hutchins will be back as our presenter as well as our co-host that day, talking about feeding for feet and legs. Zinpro Performance Products will be the sponsor that day. Again, Dr. Paul Fricke, thanks. Great job today. And Mike, are you ready with what you call the speed round?
Yes, sir. We are ready. Paul, are you ready to rock and roll here? Here we go. You got a boatload of them. How do you feel about timing AI based on inline progesterone tests rather than heat detection? Does that make sense to you?
Yeah, it does make sense to me. And I think what I showed the information on the DeLaval Herd Navigator system. One of the ideas that they're pursuing is to say, well, if we can track progesterone daily, or even every time a cow is milked, we can basically watch a cow not get pregnant, right? So she's going to have progesterone come up, and then you're going to watch progesterone decrease as the corpus luteum regresses. And what they want to do is to try to time insemination without seeing the cow in heat to get high fertility. Okay, now I am a skeptic by nature and by training, right? I'm a scientist, so the first thing I always ask is show me the data. I haven't seen that. I think they do have some data, but I haven't had a chance to look at it. But here's what I think, Mike, is I think there's too much variation among cows in the interval from regression of the corpus luteum until ovulation to have really high fertility to a timed insemination based on progesterone. That's just what I think, and until someone shows me otherwise, that's what I have said for a long time about that. But it doesn't mean that you won't get some cows pregnant. That's not the question. The question is, are you maximizing fertility by trying to do that? And I'm going to argue that in a timed insemination system where we actually force an ovulation, we can get the timing of insemination exactly right. And I don't think you're going to be able to do that just based on a decrease in progesterone.
Well, that sounds like a Democratic answer. But we'll move on to the next question. With the IDEX read test, can you run one at a time, or must all the wells be filled at one time?
Good question. The way that test is marketed is those wells are in rows. Okay, so you don't have to use a full 96 well plate, but you would have to use, if I go back to that slide, in fact, I'll just go right back here. They come in rows, so you can see column number 1, A through H, that's all together, and then you'd have a column number 2. So basically, these things snap into this thing. So you'd only have to run one group of these wells, not 96.
Do I have to run a standard curve then in every time as well if I have 1 or 2 cows to check?
No, that's another good question, and I kind of confuse people with this. This test is what we call a qualitative test. It's yes or no. Okay, so qualitative tests just have 2 outcomes, yes or no. We were trying to make a semi-quantitative test. So by running a standard curve, these 2 columns here is a standard curve, then we can express a cow as a % of a pregnant cow. Okay, so we were trying to do something a little different. I was just showing this to show what these color changes look like. The nice thing about this is you don't need to put this into a reader. You simply just set this on a white background and you just use your eyeballs to tell whether there's nothing there or whether there's something there as far as the blue color.
Very good. Thanks very much. Paul, you want to click back to our host, our sponsor again, and I'll bring you the next question. So is there a blood tests that are more popular in different areas of the United States, or any regionality to this? Do you see at this point?
It's a really good question. I don't know if I know the answer to that question exactly. What I can tell you is that Bioprin has regional labs that they've set up all over the place. In DG29, talk to your CRI representative if you work with CRI. IDEX is a national, international company, and again, those visual read tests, you could have them there. I think we're far enough into this thing where if you want these tests done, I think you can probably get them done wherever you are.
Okay, next question. What might be the strategy with a daily robotic milk testing? Any guess if this is going to be cleared for the U.S. when we might best start to use it?
Yeah, so the deal about Herd Navigator originated in Europe, and I think there's some systems in Canada. The issue comes, my understanding of the issue is that in the United States, once milk leaves the milk line, it has to be discarded. And so if you think about a parlor with one of these robots in there, the Biol Herd Navigator, you have to have tubes from every individual cow that transport the milk to that robot, and there's a lot of volume there. And if you had to throw away all that milk, it becomes kind of cost prohibitive. And so I think they're working with approval to try to get around that issue. I'm not sure where they are with that at all, and there could be people online that work with Biol Herd Navigator, but it's a neat idea. And again, I've always cited that the ability to measure progesterone sequentially in the same cow, like in milk, is going to be something that's radically going to change the way we manage reproduction.
What do you think we will use low PEGS measurements from pregnant cows to predict pregnancy loss and strategical response? And they're citing a Polar Journal Dairy Science article paper. Does that make sense to you? Want me to repeat that?
Well, okay. So if you just look on the screens here again, these are cows that lost pregnancies. They do exactly what pregnant cows do. On day 25, it's low. On day 32, it's high. And look at the variation among these animals. And then they start to lose that pregnancy. if you superimpose all these animals on top of these pregnant animals, they're doing exactly the same thing as the pregnant animals, except they're going to come down, and then they're going to come down farther and faster. So here's what I would say about that. Using a single point in time estimation of PAGs, I believe it's going to be very difficult to make those kinds of decisions. If you had serial PAGs, now let's just think outside the box, and let's think about new technologies. What if I had not only the ability to measure progesterone in line in milk, but I could also measure PAGs? So now I know high or low progesterone, and I know high or low PAGs. Now that would be a complete game changer. Okay, now I'm not convinced you need PAGs along with progesterone, but if you could do it, then you've got something, you got more information you can go on to make those kind of decisions. But just pulling a sample from a cow, I don't think you can make those kinds of decisions, because the question you got to come into, what if you're wrong? What if you think the cow's losing a pregnancy and she's just really one of these pregnant cows that dips down and she would have stayed pregnant and you gave her prostaglandin? That becomes a very expensive mistake to make.
Okay, what about this loss, 15% loss? Are you researchers asking why are we seeing this 15% loss, or is that just a biology we have to accept?
Well, that's a very good question. It's a very interesting question. Obviously, we're very interested in that, and it just ends up that there's a lot of pregnancy loss. Think about in humans. Most people have heard this. Many women lose a pregnancy before they even know that they were pregnant. That's just the kind of loss that we have in mammals. So if you consider a fertilized oocyte a pregnancy, we might have fertilization rates that range from 80 to 90%. Those go down in heat stress. But if you start out with 80 to 90% fertilized oocytes or zygotes, you're losing pregnancies all along from there. So some of them don't implant, some of them implant and don't form the placenta correctly. There's all kinds of different things that are happening. Now, what we do know affects that pregnancy loss, one of the things is progesterone. So if you have low progesterone during growth of the dominant follicle, we tend to see higher pregnancy losses between that first preg check and a later preg check than if cows don't have low progesterone or have high progesterone. So believe me, yeah, we're looking into pregnancy loss and what's going on with pregnancy loss, but it's there and you have to account for it.
So there's not much we can do about it. You just accept it at this point, but there might be something down the road.
There are some things, Mike, that we can do about it. Like I'll go back to this high-low progesterone. When we synchronize, say we use an Ovsynch type of a protocol, and we synchronize anestrus cows, anovular cows, non-cycling cows, we have lower fertility and higher pregnancy loss. So, I mean, those are the kinds of things that we know, and how would you deal with that? Well, perhaps supplementing with progesterone during the protocol. There's all kinds of different things we can think about doing. But yeah, we have to be aware of it. At this point, we're kind of managing around it.
We got two questions we just got to answer. I know we're out of time. Specificity of 95% equals 1 out of 20 cows wrongly diagnosed. What's the specificity of rectal palpation and ultrasound?
Yeah, those are totally dependent on the day post-breeding that you want to do the test. And so sensitivity and specificity of ultrasound get much higher after day 30 than before day 30. With transrectal palpation, it's much greater after about day 35 than it is before day 35. And I will tell you that none of these methods are 100%. Okay, they just aren't. So mistakes are made, or pregnant, cows are called pregnant that don't stay pregnant, for example. There's all kinds of those things that you introduce. And again, the question sounds to me that a person is a bit disappointed in 95%, and which may be right. But we did that on day 27, remember, and most of the tests, they want you to do it between day 28 and 30. And if you look at my data that I showed on day 32, if I just go back to it real quick, the sensitivity of both the blood test and the milk test were 100% and 98% on day 32. So it's all dependent upon when you do this test. And to me, that's more than acceptable right there. I was just trying to make the argument that 100% isn't necessary, but we can get almost 100% at the right time.
Okay, we'll let you go back to the back end again. One last question, and then we'll let Steve take over. Do you recommend Ovsynch or G6G as a resync program? And if it's G6G, at which days pregnant?
So that's a topic for an entire webinar. I can tell you that. I have on my website kind of what we do for resyncing cows, and what we typically do. We'll come in on day 25 to 32 and administer GnRH to all the cows. A week later, we do the pregnancy diagnosis, and non-pregnant cows with a CL finish the protocol. Non-pregnant cows without a CL restart the protocol with a progesterone insert. And by doing that and dealing with the no CL cows differentially, we end up with reasonable results to the resync breedings.
Well, I'm gonna cheat here. How high can we get pregnancy rates doing with any of the PAG tests? Just how high of a preg rate can we get using the PAG test?
Yeah, okay, so I'm not sure exactly the nature of that question. A 21-day pregnancy rate, I think you can do a fine job. Like I said, those data that I showed on day 32, whether you do a PAG test or do an ultrasound, it doesn't matter. When the sensitivity and specificities are almost 100%, it's going to be the same. Okay, so if that's the question, I think if you use the test correctly, they're great. I don't know if the question is with regard, though, to what percent pregnant cows you'd have. That's a different question. So I don't know about that.
Okay. Well, Steve, I think we're going to cut it off, and we just keep getting more and more questions here, and we still have lots of listeners. But Steve, you want to wrap it up, and we'll call it a day.
Sure will, Mike. It's showing that we have a lot of interest, and it's a good topic. Thank you again, Paul Fricke, for a wonderful job. Our thanks again to Parnell for their sponsorship of today's session. June 13th, hope you'll back with us again on Barn Designs for Robotic Milking. Jack Rodenberg from Ontario. Laley will be sponsoring, and then Zenpro will be sponsoring the July 11 webinar, Feeding for Feet and Legs. Mike Hutchins will be the presenter that day. So thanks again to all of you for being with us, and we look forward to having you join us again. And we always invite you to go to our archives or to the Hoard's Dairyman website and view archives of this and other webinars from the past. So signing off up here from Fort Atkinson today, Steve Larson here.